All right, welcome everybody. I'm Leslie Wade. I'm the programs chair for NAFEX, and I'm very excited to have you here. Thank you for joining us. And I'll introduce Chris Heater, our NAFEX president, and let Chris take it away.
Thank you, Leslie. I want to welcome everyone to this session of the North American Fruit Explorers Conference for 2022. Before I introduce our speaker for this session, I want to share a few housekeeping tips. If you've already been on, you know these things, but if this is your first session, then we'll just kind of go through a few things. First of all, you're all muted and off video. You won't be able to take yourself off. This is a webinar format. So unlike a meetings format, you guys will be remaining muted and off video, but you can interact by using the Q&A button at the bottom of your screen. So if you move your little arrow, you'll find that pop up. It's Q&A. Please use that rather than the chat. So Q&A if you have questions, and you can type questions throughout. And then as we go through, we'll share those questions with Jeff as we go. A couple words about NAFEX. Oh, I should also say this is being recorded. So if for any reason you need to jump off or you want to go back and re-listen to anything that Jeff has to say, this will be recorded and up and available on the website after you log in a couple of days after this session. So just kind of keep your eye out on the website, log in, and you'll be able to find these recorded sessions and any information that went along with the session. So about NAFEX. So NAFEX was founded in 1967. It's been around for about 55 years, a little over 55. And it was a spinoff, actually, I think, from the Northern Nut Growers. The Northern Nut Growers has been around for 110 years. NAFEX kind of spun off as a fruit-only group. It's got a very rich history. I came on in the organization about 5 years ago. I kind of hit my interest in fruit a little bit late in life, but it's been a fabulous organization. And it was founded on the principles of people getting together, both amateur fruit growers as well as academic breeders and professional orchardists, all coming together to share knowledge and to help each other. And so in that spirit, we continue all these years later. Our organization has, again, hobbyists and people who are growing professionally. We have academics, just a wonderful, beautiful mix of all generations and all different walks of life sharing information together. So I'm very excited that we all love fruit and we're all here sharing that information. There's lots of things available. So if you're a new NAFEX member, a couple things to note. We send out a quarterly 4 times a year. It goes out to everybody digitally by email, and you can access those back issues online when you log in. And if you have purchased the full membership versus the digital membership, then you'll also get those 4 issues mailed to you each year through the U.S. Postal Service. You can also access past issues of the journal, not only by reading them individually, but we also have a searchable database. Thanks to some of our volunteers a couple years ago, you can search by keyword and you can find articles from years ago that are pertaining to particular fruit or cultivar that you might be interested in. Our organization is built on volunteers. None of us get paid to do what we do. And there's lots of volunteer opportunities. So we hope that some of you will be inspired by what you see and what you hear, and maybe be willing to step up and volunteer from time to time. We're always looking for board members, people who would like to volunteer on maybe a particular project. So reach out to us at the admin@nafexmembers.org if this intrigues you, if you want to talk about possibilities. And stay in touch. We hope that you find this really useful, this particular conference, along with our interest group meetings that we record, and understand and realize that that's a great value for your $19 a year. So with that, I'm going to give a little introduction here to Jeff. So Jeff Lowenfels is an author, a columnist, and a gardener. Jeff is America's longest running garden columnist with over 45 years of continuous garden columns. He's the author of 5 internationally best-selling books, which includes a group of 4 books called Teaming With something. And I actually brought a couple of my copies. I just brought 2 of them and grab those for you. I like show and tell. So hopefully you can see this: Teaming with Microbes, Teaming with Fungi. But these are wonderful books. And actually, when I first started growing, these books were the first books recommended to me by some friends of mine who we'll hear from later on tonight. So check out those books. I'm sure you can get them at your library. You can buy them off of any bookstore website. So I encourage you to check those out. Jeff lives in Anchorage, Alaska, which is far, far away from the orchards he took care of in Scarsdale, New York. And as we learned last night, Jeff and Lee Reich went to high school together, I believe. So there's a great connection there between Lee, who we heard from last night, and Jeff today. And Jeff is considered a top comedian in the garden movement. So I don't know if he's going to elaborate that on anymore, but that's kind of exciting. So with that, I'm going to turn this over to Jeff. He's going to give a PowerPoint presentation, and there'll be lots of, again, opportunities for questions and answers. Use your Q&A box to type in your questions, and we will get to those when the time is appropriate. So with that, Jeff, I'm going to turn it over to you, and we look forward to hearing what you have to say.
Oh, thank you very much. I appreciate it. Always a little frightening to speak in front of someone like Lee, because he is perfect at this stuff. I'm just an amateur, but nonetheless, I will try to apply some of the lessons I learned from him. I am talking today about something new, the soil food web. Now, for most of you, you're saying, what's new about the soil food web? And indeed, there really is something new about the soil food web. But I got to establish some cred first. We do garden in Anchorage, Alaska. Right now, it's about 30 degrees out. It was 11 degrees last night. But yeah, we garden. And we have bees and lawns and flowers. And we've also got these guys, which look really bad. But I'd rather have a moose, even though this particular one is eating part of one of my trees, than the deer that many of you have. We grow lots of fruits, but in order to do so, because of those moose, we've got to have a lot of fencing. So we do grow fruits, and so I think I can safely say that I know a little bit about that. I do have 4 books out, but I used to have 3 books out, and so I call myself Lord of the Roots. When I only had 2, I used to call myself America's Dirtiest Lawyer. But now I've got 4. I'm going to continue to call myself Lord of the Roots. These are the original 3, and this one just came out a couple, 2 or 3 weeks ago, and it's what I want to talk about today. So let's get down to business. This is an Alaskan sled dog doing his business. Let's talk about a new soil food web discovery called the rhizophagy cycle. It's brand new. Really discovered in 2010, and I venture to guess that nobody here, maybe other than Lee, has ever even heard of this incredibly important new part of the soil food web. Now, for those of you who don't know about the soil food web, it became popularized in the early 2000s, and I wrote a book called Teaming With Microbes in 2006, for Dr. Elaine Ingham to explain what the soil food web that she had really perfected was all about. And I could give the whole talk on this slide. What happens is the photosynthetic energy causes the plant to create exudates, carbon-filled molecules that drip out of the root system into the soil. And they attract fungi and bacteria, because these fungi and bacteria in the soil don't photosynthesize. They need the carbon. They're happy right there in the rhizosphere up against the roots. And along come nematodes and protozoa that eat the fungi and the bacteria. They eat the fungi and bacteria because they too need carbon. They don't need everything that's in the fungi and bacteria. And so the excess is excreted in plant usable form. The microbes actually put a charge on these molecules so that they can get into the plant cells. And in order to get into the plant cells, which is the subject of Teaming With Nutrients, the second book I wrote, they have to have a charge on them, unless you happen to be boron. So this is what happens. This is the science behind organic gardening, and it's called microbe-mediated solubilization of soil nutrients. And again, what happens is the nutrients are released from all the eating, and they travel through the soil into the plant. This is the exudate back in 2006. This is the best picture I could find. Here's a picture today that shows a root, some of the sloughed off cells at the tip of the root, exudates, and the attraction of bacteria. These are individual root hairs. So let's show you how this works. This is a tree sitting around in Maryland. It decides it wants Japanese food. Its neighbor wants Mexican food. Its other neighbor wants French food. But because it's bigger, it gets the Japanese food choice. And so what happens is it develops, these are the roots of that tree, let's put them so that they look like roots. It develops an exudate that's designed specifically to attract Japanese bacteria, which it does. And if the plant wanted something other than Japanese food, say it wanted American food, it just needs to change the exudate, and lo and behold, it ends up with American food. The plant is in control. We like to think we're in control, but in fact, left alone, the plant can do just fine without us. So what happens again is these microbes are putting a charge on these molecules that travel through the soil. This is the system that Dr. Elaine Ingham explains. This is the system that we all know about. And again, it's based upon the fact that you've got to have a charge on these things in order to get them into the plant. Now, in 2010, mycorrhizal fungal symbiosis was really added to the soil food web picture of 2006 because people were able to start to grow these in labs. They became available at nurseries, and 90% of all plants that was discovered have a relationship with a mycorrhizal fungi. There are about 350 of them. We know how to make about 15 of them, and about 8 of them are commercially available. And these fungi go into the plant. This is a plant root. The fungi are much, much smaller than the plant root. They're capable of sitting in between the cells of the plant root where they collect the exudates, and in return, they bring back and distribute into the plant all sorts of great nutrients. And because they're so small, they're able to get into pore spaces that the root itself wouldn't be able to take in. Or to get into. And they are very responsible for lots of the phosphorus and the nitrogen, zinc, copper, iron, you can read as well as I can, for getting into plants. And here's how they operate. So you have two seedlings sitting around on a sunny day, and one of them says, I would really like a bologna sandwich. And so it goes into its mixtures, mixes up a bunch of stuff and exudates it out. Lo and behold, it catches a mycorrhizal fungi. The mycorrhizal fungi gets the bologna sandwich, brings it back to the seedling, and then continues to feed the seedling so that the seedling grows. And what's really cool about it is that often plants next to each other will share the goodies that their mycorrhizal fungi are collecting, including water, I might add. It's very, very neat. So that's the second way that plants get nutrients. And then finally, we have what I really want to talk about, which is the endophytic bacterial symbiosis. Endophytic simply means that the organism lives a part of its life inside the plant. Endo, in, phytic, plant. Inside the plant, and it helps the plant, or at least it doesn't harm the plant, and it doesn't spend its entire life inside the individual plant. So that's called endophytic bacterial symbiosis, and it has a very special kind of bacteria, also some yeast, but mostly bacteria, that are endophytic and engage in something that you've never heard of called the rhizophagy cycle. Rhizo meaning root, phagy meaning to feed on. It was discovered in Australia by this young lady here. I won't even try to pronounce her name. She and her grad students used confocal microscope, and they saw that bacteria were being engulfed by root meristem cells and were doing something inside the root cells that nobody had ever seen. They seemed to be feeding the plant from inside the plant. She called it rhizophagy. She actually called it rhiza something or other. She pronounces it in an Australian way, but the American way to pronounce it is rhizophagy. Her grant monies ran out. And unfortunately, when grant monies run out and you work at a university, you have to move on to something else. But fortunately, a gentleman named Dr. James White picked up this work. He's at Rutgers University, and he and his students have been in the forefront of rhizophagy studies ever since. So this was 2010. Now, we're talking about bacteria. And it's very important to understand that we're not talking about a single bacteria. Bacteria don't exist as singles. We're talking about massive numbers. When you say bacteria, again, it's not a single bacteria, a bacterium. It's the head of a pin with bacteria on it. Unbelievable numbers. And that's because bacteria multiply every 20 minutes. This is a very, very recent picture that appeared about 2 or 3 weeks ago. This is a single cell of a tongue. This is not the whole tongue, but this is just one cell. And these are all the bacteria that have attached themselves in the bacterial slime onto that one single cell. These guys are plentiful. They can be really beautiful. They're awfully small. This is a microscopic picture of a special kind of rotifer in the soil moisture film. And if you look closely, this is a fungi hyphae. And if you look closely, you can see these little teeny bacteria on them, incredibly small. And even though they're small, they're plentiful, and they produce phytohormones, plant hormones, gibberellin, auxin, cytokine, all the things that you know about for development of fruit, dropping of leaves, seeds going into dormancy, breaking dormancy. These are things that phytohormones that bacteria are capable of producing. And they're capable of sneaking into a plant. And plants are full of bacteria. The basic endophytic bacteria get into a plant where the roots begin to branch. There's always little cracks. And so they get into the phloem, xylem, they move throughout the plant, they can get in through stomata. And why would a plant let a bacteria in? Well, that bacteria must be supplying something for the plant to have evolved to allow a potential enemy into the plant. And lo and behold, there are specific relationships between specific plants and specific bacteria based upon eons and eons of evolution for that to happen. Now, the bacteria cause the plant to stiffen up, to increase its systemic resistance when they enter into the plant. And so they help the plant in many different ways, giving these phytohormones, but they also increase stress tolerance in these plants. And so it's a very important thing. Very few people study this, but in fact, more and more are studying it because of what I'm about to tell you. So here's the stomata. These are the bacteria that are jumping into the plant. There's some more over here. But let's talk about the rhizophagy cycle. So we've got your root, and you got the root cap. You've got the meristem cells. Now, these meristem cells, and then you got are very, very thin-walled. They begin to stretch as you get into the area of cell elongation. And then, of course, they begin to form root hairs. What happens is that the bacteria, and this is a good depiction of that. This is the root. These are the root hairs, exudates, bacteria. What happens is the bacteria literally are taken into or attracted into the plant cell. So let me show you how this works. Now, you remember those, and incidentally, this is what I'm talking about here. So this is the new kind of microscope. Had Dr. Elaine had these microscopes, I'm convinced she would have known this. If you look carefully, you'll see that in all of these root hairs, there are bacteria. This is where the bacteria enter. This is the area of elongation, and here they are in the root hairs. Now, some of those bacteria that are attracted to the rhizosphere by the exudates of the plant are not eaten. They're stubborn. And in fact, what happens to these guys is they're sitting there happily eating away in a nice bacterial colony with a slime around it, and they begin to smell butter. They begin to smell buttered popcorn, butyric acid, which is being released by the plant. They're in this slime, and they're competing with all of their other cellmates in that slime, and I mean cell by terms of jail, and the smell causes them to say, there's an area outside of the slime I can go to that has less competition, and I'm going to do better. And so it breaks through that very thin meristem cell wall in the root, and it finds itself, once it breaks through, going through the cell wall in a place called the periplasmic space, which is this little area around the cell membrane. And the cell membrane, of course, contains all of the organelles in the cytoplasm of the cell. Let me refer to a typical tofu container as a potential way to understand this. So outside the tofu container, that little white outside and this plastic here, that's the plant cell, the meristem cell wall, very thin. And the bacteria poke their way through that and find themselves in that liquid that is always around that tofu, which represents the membrane held cytoplasm. So in this space here appear these bacteria. And when they appear, and just so you can see what it looks like, this is a meristem cell. These are the bacteria. They line up in that periplasmic space, and they're circulated around as a result. And here's a closer up picture. And they're circulated around that periplasmic space. Now, when they go through that cell wall of the plant goes, uh-oh, wait a minute, I'm being attacked. So what it does is it sprays the entering bacteria with something called a reactive oxygen series. Hydrogen peroxide is a very weak reactive oxygen series, but this is like superoxide, they call it, a reactive oxygen series, ROS. And what it does is it sprays the bacteria and removes the wall of these tiny little bacteria, and the goodies that are in that wall are absorbed by the plant. So ROS, reactive oxygen series, incredibly important. When you get sunburned, the reason your skin turns red is because it's reacting to a reactive oxygen species that is produced by your skin cells. And I even found a ROS that was sunburned, which I thought was kind of cool. Anyway, what happens is the ROS strips the bacterial cells off. Now, this ROS is also spraying the plant cell wall. And so again, the plant has to fortify itself to be able to handle this reactive oxygen series. And so a byproduct of the bacteria entering is a strengthening of the plant itself. Now, the bacteria do not like being sprayed with ROS, and so they fight back by spraying the bacteria, or by producing in the periplasmic space ethylene. Ethylene is a phytohormone, and it causes plant cells to stretch, which is also another thing that it does is it mixes with carbon dioxide and water and forms carbonic acid, which weakens the ROS, sort of an antioxidant, so that the ROS isn't as strong and more of the bacteria isn't destroyed. So the plant cell stretches because this ethylene that the bacteria that just entered the cell produce cause the cell to stretch. That's the phytohormone working. And when it stretches, a little bit of carbon leaks out into that endoplasmic space, periplasmic space, and that carbon is used by these wall-less bacteria to continue to grow and live. Now, they also produce nitrite. They fix nitrogen inside this cell, and that nitrate is converted to nitrates and absorbed by the plant. They're producing the nitrate, again, in order to weaken the ROS. And the bacteria spray that out, it gets converted to nitrate, and it's absorbed into the plant. Unbelievable, because nobody really knew this was happening. The microbes are trapped. If they want to get that carbon in order to be able to live, they have to produce the ethylene. If they want to save themselves from the superoxide, they have to produce the nitrogen. And so they've got these two loops going on. And all the while, they're being circulated around the plant. And all of the nutrients that are being produced by all this activity are eaten by the plant, which is really kind of neat. Now, their circulation continues, and so does the multiplication of these bacterium. And they are circulating around, multiplying like crazy every 20 minutes, and you end up getting these blobs, these little 4 or 5 sets of bacterium. They look like little beach balls, and they multiply and multiply throughout the root meristem cells. And they're circulated, and the ethylene is circulated, but they're multiplying and multiplying. And what happens eventually is you get so many of them that they begin to back up against the plant cell wall. And the ethylene that they're producing no longer gets circulated, but is concentrated in one area. And when it's concentrated in one area, it causes that cell wall area to stretch, to form a tube. And so you've got your basic meristem cell, you get the bacteria backing up, and they form a tube. You and I know this tube to be a root hair. If you don't have bacterium, rhizophasibacterium, working their way in these cells, you do not get a plant with root hairs. If you put the bacteria back, you do get root hairs. Absolutely amazing. This is the root hair system, and you can see all of them have bacteria in it. You and I were taught that root hairs were there to absorb nutrients. They do so only because the cell walls are so thin. But these hairs are there not to absorb nutrients, but to deal with these bacteria. So the bacteria are multiplying. They form these or cause the root hairs to start to grow. This is a beautiful picture. You can see it's all one cell. So this is the root hair coming out of this cell, and bacteria are forming into it. What happens sometimes is you literally get a tidal wave of water that pushes the bacteria into the cell, up towards the tip of the cell. And again, here pictures of the root hairs with the cells, with the bacteria in them. And they're stained, their metabolites are stained here so that you can see these blue indicate presence of the bacteria. And they form at the tip, they get more and more and more forming at the tip. And then finally they're shot out of the tip back into the soil, where they regrow their cell walls. And then after 2 or 3 days of living in the soil, if they're not eaten, they go back into a new meristem cell and repeat the process. And when they're in that root hair, they don't just blow out once. They blow out, as the root hair grows, several times. 1, 2, 3, 4, 5, 6, 7 times sometimes. They just blow out all of these cells that are wall-less. They blow out the tip into the soil, where they regrow their walls, renutrify themselves, and then repeat the trip again. So again, here's a root hair. Boom, 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 boom. These root hairs are not there to gain nutrients. They're there to blow out these bacteria. So under the 2006 version and the 2010 version with the mycorrhizal fungi, really what we've got is a plant that's a farmer. it puts exudates out, the crop grows, the crop, fruit is then dropped to the ground, and it moves into the plant. What you've got now in the 2022 model is the 2006 system, but you've also got a rancher. So the plant is a rancher as well as a farmer, because what it's doing is it's taking in the sheep, shearing off the wool, eating a couple of lamb chops, and then putting the sheep back out the pasture so that they can regrow their wool and bring them back in again. Wow. Completely new part of the soil food web. So these particular bacteria, they work, they save, they travel, they repeat. And this is a depiction of what the soil food web now also includes, the rhizophagy cycle. And it's an elegant, beautiful cycle. In the book, Teaming with Bacteria, which describes the cycle, are some of these unbelievable pictures. I would recommend that everybody put rhizophagy into their search engine so that as Dr. White and his students and other people around the world, China's beginning to look at this very carefully, can keep up with the activity. Because what's happening here is that you've got up to 40% of the nitrogen a plant uses coming from the rhizophagy cycle. That's a lot of nitrogen, and it's nitrogen fixation inside the plant, which is of keen interest because of the implications. Bayer and all the big companies that make fertilizers, chemical fertilizers, are moving into the biofertilizer situation. They're going to be studying and developing, and they are studying and developing, microbes that fix nitrogen inside particular plants. So it's coming, it's here, and it's pretty darn exciting. So again, this is how they get into the system right in here, the rhizophagy, and then you've got the other kind, the other endophytic bacteria that get into the root cracks. And let's just talk about them a little bit. They go in, there's plenty of food. So if you're asking yourself, why would a bacterium want to go through the process of taking the chance of being sprayed by Ross and killed, it would do so because inside the plant there is less competition, ready food source, and sometimes the plant even helps the bacteria because it's so important to the life of the plant. Eventually, these endophytic bacteria end up inside the flower, and they get caught up inside the seed as the seed forms. And then when the seed is germinated, the bacteria that are contained jump into the soil and help feed the plant and help establish the populations that the plant needs for the rhizophagy cycle, for other endophytic bacteria to move in. This is the source of these bacteria. I mean, ultimately, it's the soil, but they get caught up in the plant system, and some amazing things come out of this. So, for example, a 400-year-old strain of corn grown today has the same bacteria mix, basically, that it did 400 years ago. It's incredible that these bacteria seem to be imparting very specific traits to plants that we might call terroir or associate with terroir, the soil, but in fact it's the bacteria. So they're doing all sorts of great things. The reason why a plant would let them in again is because they're capable of doing some of these things and helping the plant. So over the years, eons and eons and eons, individual plants and individual bacteria have formed these symbiotic relationships. And unlike the mycorrhizal fungi, which tend to sleep around, one fungi can infect 200 different kinds of plants, not necessarily so with the bacteria. And so this is why it's being studied so closely, so that we can find the right bacteria for the right plants. And again, these are the things that it does. It helps the systemic resistance that's created when they enter into the plant, helps them handle abiotic stresses and biotic stresses, all incredibly important. And they can do things like change the temperature. In fact, you can even buy now a mixture that you can spray on your fruit trees that changes the way water freezes in the cells of the plant. And that's what's happened here. These contain an endophytic bacteria, that this lawn doesn't have. I'm going to go back. Whoops. I like to think this is the organic lawn and this is the one that uses all the bad stuff. And these bacteria are capable of producing things that can hop out into the soil, cytophores that can tie up metals so that they don't poison a plant, or can grab a metal that a plant needs and bring it into the plant. There are some of these bacteria, endophytic bacteria living inside the plant, will jump out of the plant and take out a bad fungi, or work with a fungi to develop enough phosphorus for the plant to be able to use. All important stuff. And some of them are already available for use by growers. So these on the top, this is a nitrogen fixator. I'm not going to even try to pronounce the names. Azospirillum, I can say that. These are all available at really good nurseries and actually grow stores, the place where cannabis growers go. This particular one has to be freeze-dried. It's becoming available, but these are definitely available, and they work on certain plants, not all plants. And so people need to start experimenting with these to see whether they grow on your particular fruit. So there's some pretty obvious takeaways here. The first is that the plant's in control in ways we might not even know about, which is why it's so important to try and let plants do what they do naturally without your interference. They're absolutely amazing, as you can tell. They're beautiful. These are beautiful bacteria, but they're all different. And they all react differently with different things. We need to start doing the research to make sure we know what goes with what. So you don't sterilize seeds because you kill those bacteria. You don't sterilize the soil. You don't toss used soil because that used soil contains the exudates and a lot of the bacteria and other microbes that you would want to grow your tomatoes in. Why are you throwing it out? If you don't have a root problem, reuse your soil. And of course, you want to buy a copy of Teaming with Microbes, not so much for me, but because it contains 19 rules that everybody should be following to preserve the soil food web. There was a 20th rule. Timber Press took it out. They didn't ask me, but they just took it out. The 20th rule was never lend a copy of Teaming with Microbes to anybody. Make them buy their own. Timber didn't think that was funny. Anyway, chemicals have a very negative impact on endophytic bacteria. And when you use chemicals, you're impacting the rhizophagy cycle. You're impacting the ability of these plants to produce nitrogen internally as a result of these bacteria. This doesn't change anything with regard to the use of chemicals. It just adds another reason not to. And of course, rhizophagy and endophytic benefits include the growth and the formation of soil structure. The slime that's on these bacteria sticks to a particle of soil, which then sticks to another particle of soil. And because they're not flat, all these particles stick, and fungi come in and weave them together. They're not flat. And because they're not flat, you've got pore spaces where air can go in, where the little guy can hide from the bigger guy. This is where soil structure begins. And if you do anything that impacts the bacteria, endophytic, you're doing bad things to soil structure. We're told we only have 60 years left of this soil because the processes that people use, the commercial farmers, destroy the bacterial situation. And as everybody knows, we don't rototill, we don't compact, we treat our soil very carefully. So some of the not so obvious takeaways, we need to take a look at your individual fruits. It appears that my heirloom tomato, for example, and you can extrapolate out into the apple trees and whatnot, my heirloom tomatoes are different than Lee's heirloom tomatoes, same seed, because I've got different bacteria. Tomato heirlooms develop as a result of these specific things. It turns out that the compost that's made out of vermicompost, worms, has a different bacterial base than thermal compost. And going back to what we now know, that some bacteria can help some plants, we need to experiment and use vermicompost and check it against thermal compost to see for which individual plant one is better than the other. Which one? Or is it a mixture? And I can see this work being done now and being shared next year at one of your meetings. We make compost using the same parts of the same plant and use that compost on the same plant. You tend to get the bacteria that plant needs in that plant. Best practices, well, you all know what they are. Obey the law of return is the key one. Feed the soil. And what we do as fruit growers is we remove the fruit. And that would have rotted underneath the tree, and the goodies there gone back into the soil to feed the bacteria so that they can feed the plant. But we remove that stuff. So instead, we need to be using living mulches, which are all going through the rhizophagy cycle, and wood mulches, compost, soybean meal. From my perspective, if you want more bacteria, then you chip up that wood mulch and make it very fine, and it checks more bacteria. And you want to monitor your nutrients. When I give my talks, I talk about a product that I advise the company. It's called a microbiometer, www.microbiometer.com. What it does is it measures the biomass in your soil. And so if you apply an organic microbe food, you should be able to take a test when you make the application, and then take a test 3 or 4 or 5 weeks later to see whether it's actually helping or whether it's just simply doing nothing. This is really kind of a neat thing, www.microbiometer.com. And you need to get NPK tests as well. I mean, you do need to know what's going on. Don't disturb the soil. That's the biggest takeaway. You feed the soil and you don't disturb it. You prevent compaction underneath your trees. You don't till. You aerate if your trees are compacted, and you tell the kids, don't play underneath the trees and don't climb in them. And of course, one of the best practices is to buy the Teaming series of books. I would start with Teaming with Microbes. I would then go to the new one, Teaming with Bacteria, then Teaming with Fungi. These explain how the nutrients get to the plant. This one, Teaming with Nutrients, the last one to read, explains what happens once the nutrients get into the plant, how they get into it, and what happens once they get into it. So if you're not convinced, I always say to people, just think about this. Whoever fertilizes the redwoods? How'd they get to be so big? How'd they live so long without fertilizer, without Miracle-Gro, without herbicides and pesticides? They do so because they team with the soil food web, and they've known for a long time, and now you know that part of that teaming involves endophytic bacteria and the rhizophagy cycle. So there we go. Thank you very much. If you've got any questions, you can certainly reach me here. And by the way, don't forget to harvest something in your garden for the hungry, and next spring, plant a row.
Wonderful. Thank you, Jeff. That was amazing. We've got some questions, and so I'm going to kind of go through these, and then if time permits, I've got a couple questions for you too. So Andrew asks or says first, something that seems to have become quite popular of late is the charcoal soil amendment. It seems to be quite beneficial, but few can say why. Do you think it increases the carbon supply in the soil and therefore feeds the microbes you're referring to?
Yes. If you charge you've got to charge it, and that's putting the microbes into it, the biochar. And there's no question that biochar is a condominium for microbes, and so that's why it works so well. It's interesting that we all talk about this. The biochar in terms of South America and how it was formed out there. But if you wander around the Northwest, you see that the natives in the springtime would grow little teeny potato-like things, and then they would burn every season the soil to create the carbon, which then got charged during the year, and then next spring it was used again. So yeah, definitely.
Great. So Juan Carlos asks, and I'm going to kind of preface before I ask this, he's talking about bacteria, and he wants to know how long these live when you're talking about them entering in, dividing, and then shooting back out. And just to remind folks, bacteria typically in a laboratory setting at 37 degrees Celsius will divide every 2 hours. But in this case, how long do these bacteria live? And I guess we need to keep in mind that they're dividing while in the root, and then shooting back out. So in essence, how long do they live before?
Yes. Well, they spend about 2 days in the root meristem cell. So for 2 days they're dividing, so obviously some of them are, so 2 days in the cell and 2 or 3 days in the soil before they go back in again. Okay. And don't ask me how they figured that out. I don't know. This fellow, Dr. James White, is good. Dr. Elaine Ingham is good too. And together, wow. They really have changed the picture. But so they last a couple of days. How long does a bacterium live? In theory, forever. But it's always multiplying. So,
right. Yeah, it's always multiplying. So, divides, and then, maybe one of this ancestors dies off, but it keeps dividing. So as long as there's
or it goes into a spore situation like some of these bacillus do, and that's the reason why those 3 or 4 ones that I listed are available for sale, because they go into a spore form, and so storage of them is relatively easy. They'll store for 2 or 3 years, whereas if it's a live bacterium, as people know, it's a difficult thing to handle, and shelf life not so good.
Yeah. All right, let's see. Matt asks, how do endophytic fungi fit into the overall picture of the plant?
Yeah, there are endophytic fungi just like there are endophytic bacteria. Of course, the mycorrhizal fungi are endophytic. They're living inside the plant for part of their life. But there are actually some yeasts that go through the rhizophagy cycle. And interestingly enough, as the bacteria divide, the regular bacteria, not the yeast, inside the periplasmic space, they form something called L forms. That's what they call them. A wall-less bacteria is called an L form. And the only reason it's an L form, incidentally, is because they were named at the London School of something or other, and so they put the L in there. The L forms bleb, like yeasts do. Is that the right term? Bleb. You know how a yeast will just make a little piece of, but yeah. And so that a lot of that happens during the rhizophagy cycle as well. The pictures are unbelievable. And I very rare do you see this in a book, but I have in the back in the appendix a website where you can go and actually see the root hair growing and moving, and it gets better and better and better. I can only imagine, what Dr. Elaine would have done with a confocal microscope.
Yeah,
pretty amazing stuff.
The technology has significantly improved in the last 10 years. So, it's amazing.
Yep.
All right. Let's see. Regina, with all the earlier focus on mycorrhizal fungi, what significance does this new information on bacteria have for us as growers?
Well, I don't think it detracts at all from mycorrhizal fungi. They're still very viable. You want to use them. I would suspect that a lot of these endophytic bacteria end up working with the mycorrhizal fungi, helping them fix phosphorus, et cetera, et cetera. So there's a synergism between the bacteria and the fungi. You'd have to look at the individuals in order to know how big it was and what it was.
Yeah, it's almost like it's just another piece of the puzzle, right?
Right. This does not wipe out the old soil food web. Dr. Elaine is still the guru of the soil food web, as far as I'm concerned. This is a splay that's added to the soil food web. Yep.
Let's see. Dr. John Cummins asks, Do you think these cytophores might be able to mitigate against the harmful poisons being spewed out by governmental chemtrails?
I don't know. I have no idea. I don't know how you get them up there. Obviously, the chemtrails fall to the ground. I don't know is the answer.
Okay. And so Dan Lefevre adds as a kind of a reply to Dr. Cummins' question. He says certain ones may be able to sequester and change chemical form to a more inert form. Humic acid can also chelate and lock up some heavy metals.
Humic acids. There's no such thing as humic acid.
Yeah.
They're acids. Yeah. Definitely. I've been using humic acids in soil for a long time. They're really incredible.
Yeah, and I would imagine. I mean, you talked a lot about the roots, the bacteria and the root symbiosis and everything, but there's also bacteria on the leaves, and in our cases, trees, trunks of trees, and things like that. So perhaps there's a lot of things going on there as well.
Yes, there definitely is. So, for example, let's take trichomes, which are on leaves, cannabis, tomatoes. A number of plants have trichomes, and what people thought originally was that the rhizophagy cycle was causing the trichomes to form, just like the root hairs form. But in fact, what now is being established is that they fix nitrogen inside. The bacteria can fix nitrogen inside those trichomes. In order to do so, they need to be in an oxygen-free environment, or at least a heavily reduced oxygen environment. And in the case of cannabis, it's the plant cells spraying cannabinoids into the area so that you get an oxygen-free area, so nitrogen can be fixed in the trichome. Very interesting, and a lot more research being done.
Very cool. Andrew asks, can you suggest any commercially available bacteria that might be beneficial to apple trees specifically?
I can't. But again, if you go to these growth stores, the reason why I thought this would be an appropriate talk for your group is so that you folks can start to experiment. We need to find these answers out. And in 10 years, we're going to be able to take our cell phone, I'm convinced, and be able to take a little mixture of water, because this is what that microbiometer does, and use our cell phone to read the RNA mixture and tell us what bacteria we have in our soils. It's coming. We've got the databases. We just don't have the instrument that you and I can use, but you and I can send a sample of soil into somebody and pay a lot of money, and they'll tell us what's in that soil. We're getting awfully close, but it's time for us to be doing these experiments, just like we did with the soil food web when it first came out. Lee and I would go back and forth about compost tea. Had to experiment. And that's what this is all about. And that's one of the great things about an organization like yours, is that you've got the capability of conducting the experiments and then sharing the information.
Yeah, I agree. Dr. Sandy, she's an expert in the nut world, but she says she loves this talk. Even old mycologists can learn new things. So passing that on.
Well, I'm honored, because let me tell you, I did not invent any of this stuff. Same with the first book, Teaming with Microbes. I am a lawyer by trade. I'm not very smart, and so in order to understand this stuff, I had to dumb it down so that I could understand it, and hopefully, students and general public people are going to be able to understand it as well.
Yep, excellent. Janet asks, does dormant oil spray adversely affect the root bacteria?
I would imagine it does. But again, let's do some experiments. Yep. It could help it, I guess. I guess in theory, some of these bacteria could eat that dormant spray and be very happy. It could hurt it. And it could certainly hurt the endophytic bacteria that are on the leaves and other things as well. On the other hand, if you're spraying because you've got a serious problem, you got to weigh it all out. You got to weigh it all out. And although Lee may be upset about this, if you've got to use a chemical, make some compost tea out of your compost and put it back in again, spray your leaves, etc. So.
Yep. Dr. John Cummins asks, what about chickens and ducks under our trees? How do you feel about that?
I haven't read. I read one paper. I read one on steer. It seems to be beneficial. They don't cause a heavy, serious compaction. Their poop eventually is usable. So I don't have a problem with it. Again, maybe we need to do some serious experiments, but I would imagine it enhances the bacterial mix.
Yep. Let's see. Stephanie asks, what are the implications of mono versus diverse polycultures in the rhizophagy as well as crop rotation?
Yeah. If what you're asking is there one kind of bacteria in the rhizophagy cycle per time? I think there is. I don't think you get 5 or 6 or 7, 8 different kinds of bacteria in. If the plant has developed a relationship with the bacteria mixed in the soil, and it only lets the one that it wants in. It doesn't let everybody in. It's not that easy. And then what was the second half of the question? You want to have a polyculture in the soil. Absolutely.
Yeah. I think she might have been talking about growing a monoculture, like a one crop versus a polyculture crop.
Well, I think it probably will depend on the crop. And I think you probably get benefits for both. And depending on the particular fruit in question, you have to determine what benefits you ultimately want to have.
Yeah.
Again, so much experimentation. There's some great theses here. I mean, this is ripe stuff. And really, I keep saying I don't know the answer. We have spent, Lee and I learned how to garden from our parents. And they learned how to garden from their grandparents. And we're just passing on mythological stuff. And we're trying to get a little bit more scientific about it. And now, in order to do that, we need to do the research that our parents and grandparents didn't do. They just accepted things. And so we got to find out.
Yep. Josh Footer says, thank you, Jeff. Our master gardeners in Cherokee County, Georgia, still participate in a plant a row project.
Great. The best. That is really a terrific program. For those who don't know what it is, you take one row in your garden or one fruit tree, and you dedicate it to the hungry. You harvest it. You take it to someone that needs it. Nothing slips between the lip and the cup. No administrative fees, no government funding, blah, blah, blah. Great program.
All right, let's see. Bradley says, you mentioned using compost from the same plants we are applying them to, but fruit growers have been conditioned to dispose of pruned material for sanitation. Could you give some thoughts about that?
Well, I don't have enough fruit trees to be able to do experimentation, but I've experimented on my crops. And I get much better tomatoes, for example, if I continue to use. Same thing with cannabis, frankly. So again, we need to experiment. We just need to experiment.
And I would add I think it was Bradley who asked that, be sure to check out the session tonight with Robert and Juan Carlos from Sober Mesa. Some of the things that they're doing in their organic permaculture system with fruit trees kind of addresses this a little bit. One of the techniques that Juan Carlos in particular is using is from South America, where they're using soil from trees that are growing really well and amplifying the microbes from those trees and putting them on the same tree. So, for example, persimmon. If I give him soil and the leaves and stuff from underneath a persimmon tree of mine that grow really well, then he takes and amplifies that and puts it under his new young persimmon tree. It just provides those beneficial organisms that I know my tree has. So he'll talk a little bit more about that tonight.
Yeah, and that's in fact the way I believe Dr. Elaine's thesis was, going around finding the area. This is the best banana in the world. That soil microbiology can be, a database put together on it. So, and rhizophagy and endophytic bacteria play right into that, right into it.
Let's see. So Sarah asks, does this cycle pertain to traditionally plantless systems like cryptobiotic crust?
I don't know. The way I approach it is if a plant has root hairs, it's going through the rhizophagy cycle. And I haven't taken it beyond that. I probably need to. But I don't know. Is there a periplasmic space? Is there? You probably know is the answer. There are no root hairs.
Yep. Let's see. Molly, going back to.
But let me just add one other thing to that you might find interesting. Dr. White discovered bacteria in places in plants that nobody ever thought they'd see them. For example, inside the nucleus are bacteria. Nothing gets inside a nucleus. Nothing, because the nucleus won't let it in. But there are bacteria in there. Are they producing nitrogen, fixing nitrogen that's being used for DNA? I don't know.
That's wild. That is, yeah. Let's see. So Molly, going back to the biochar-ish question she had, what happens if one doesn't charge the biochar?
It has to charge up, otherwise it's not a condominium. It's a condominium, but it's an empty condominium. So you get carbon that's there, and eventually they're going to be bacteria and fungi and other things that are going to move in. But you want to get its use right away. You charge it, and you get it working right away.
Yeah. So think of it as a hotel. If you don't put people in the hotel right away, it's going to take a while for people to find the hotel and do the job that it needs to do.
Right.
Dan Lefevre says, excellent presentation and kudos to whomever brought you on for this. John says, why use the few species commercially available bacteria when we can inoculate with biodiverse native topsoils and others? I think we've kind of talked about
yeah, it probably makes sense to do the latter. But if you want to go buy something, if you're a home gardener and you're not into this stuff, we want to make this so that people aren't using chemicals. And so we want to make it so that, Aunt Sally and Uncle Bob, who don't want to listen to a presentation like this, just want to have a nice tomato plant, can go and buy this stuff and put it on just like they can that stuff that angry Scotsman is yelling at us to use every spring. So.
Great. Lyle asks, does compost tea help add sugars to soil to facilitate mycorrhizal generation?
Probably not.
Nick Casco, one of our board members, asks, have you heard of cordyceps, a fungi that parasitizes ants, kills them, and grows a mushroom from their body to release the spores? Would this be a good way to control ants in the future?
It sure looks that way, and I love those pictures. But again, someone's going to have to experiment, and I'm sure they are experimenting like crazy. Paul Stamets came up with an ant fungi that dealt with an ant. And I'm quite sure that these things are being studied like crazy because it's just so cool.
Yeah, very cool. Juan Carlos, who's going to be one of our speakers this evening, and he's actually the one who told me about your books years ago. And I think he told me the other night it was one of the first books they read. He said, I need to thank you for helping us to understand the complexity of life. You got me inspired to learn more and more about microbes and our great ancestors.
I will send you my bank account after the show.
Let's see. He said also, but something very important is that all the research you do, it's passed on in a simple way to farmers, and we need that.
Thank you. But again, I did not invent it. The credit goes to Dr. Elaine and Dr. James. All I am is a lawyer that just needed to figure out what they were talking about.
Let's see, a comment from John. If the native soil is unsuitable for fruit trees, like in the northern boreal, is tilling not a good idea initially to mix the soil to provide a charged medium to allow a new bacterial environment that encourages more suitable conditions?
Yeah, it depends on what the problem, what's preventing the fruits from growing. So, for example, in the prairie, trees don't grow. But if you change the fungal bacterial mix, you can grow a tree in the prairie. And so it might be simply a need to change the fungal bacterial mix, which might not require tilling. Tilling would clearly put the system into a much more bacterially dominated system, which might be what the plant needs. I don't know about the particular plant. So, you've got to take a look at it from that regard.
Yep, I agree. I have a couple of kind of obscure questions. So you talked about the corn, the 400-year-old corn seed, and the bacteria that it brought with it. Any idea if anyone's looking at the genomes of those bacteria to see how closely they're related to what we see today?
Absolutely. And they're doing it on lots of different crops. Again, cannabis comes to mind because people are studying it for obvious reasons. It's going to be a big, it is a big business, but it's going to be bigger. And what they've discovered is that when you grow cannabis, the soil mix seems to be pretty much the same in terms of the bacteria, but in the endophytic mix is different. And it seems to be that endophytic mix which determines individual strain characteristics that people are going after. It's pretty interesting.
And kind of on the business side of things, so, I think a lot of people in this audience are leaning toward organic and more natural ways. Do you foresee or have any concerns about industry trying to breed, whether through natural breeding or through some of the more non-natural things like CRISPR, trying to breed varieties that are better at this rhizophagy?
Yeah, I am a little bit concerned. I don't know enough to be fully concerned. I guess that's probably the right answer. Anytime you try to manipulate nature, you have the opportunity to really screw things up. Yeah. So far, what people are looking at is this nitrogen fixation situation. How do we grow wheat without having to pour tremendous amounts of chemicals on it? I think people are looking at some pretty simple, benign things, and hopefully everything will come out okay. But yeah, so there's always a problem when you're dealing with nature like this, or potential.
Yeah. I mean, I always, as a scientist and seeing how things are funded right now, there's more grant money coming from companies sometimes than through the older, government grants where researchers had a little more ability to choose what they studied. And so
sure.
Our researchers.
Yeah, and it impacts people, I would not imagine the chemical companies are really that excited about a university spending a lot of time studying rhizophagy.
Yeah. Let's see. Pat asks, how do you deal with the toxic forever chemicals in the soil?
It's tough. If anything's going to be able to deal with them, it's going to be the bacteria and the fungi and the microbes. I can't think of anything else that's going to be able to deal with them in a better fashion. Again, we're going to have to figure this all out. We better do it quickly because we don't have that much time.
Yeah. Dr. Sandy says, I'm fond of ants because of the fungal spores that they carry around. Carpenter ants carry chestnut blight spores, including those with the biocontrol virus. And for anyone who's not familiar with this, there was just a recent podcast that Dr. Sandy was on talking about this biocontrol virus. So she says, please don't kill the ants.
Yeah. Oh, no, don't kill the ants. Ants are great. Ants also create soil structure. They bring organic material down into the soil. When they die, they become food for the soil food web. Ants are terrific. Even though occasionally they may bite you.
Yes. Matt says, you mentioned the soil microbes add charge to nutrients for plant uptake. Is there anything one can do to support this process of charging soil nutrients?
Just have lots of microbes.
Let's see. Bradley says, you continue to reference herbaceous plants like tomatoes and cannabis. I think of those plants as thriving more in a more bacterial soil, and woody plants thriving more in a fungal dominant soil. Is that the case? And I would personally say yes, and I think you would too.
I would too, yes.
Do the bacteria inhabit woody plants equally?
My understanding is that if you take the continuum from, the beach to the old growth forest, when you start out, you have a set amount of bacteria that basically stays the same through the continuum. What you add are the fungi. As material drops off the plant, fungi come in to eat it, blah, 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 blah. So the bacteria number basically stays the same. I think a lot of times people think that because it's an old growth or a woody tree, it doesn't have bacteria. It does. It just doesn't have the high ratio.
Yeah. So the ratio just shifts, just more fungi to bacteria.
Right.
Let's see. Juliana, what do you think about EM, effective microorganisms, a Japanese product?
Yeah, and we might as well throw in KNF, Korean Natural Farming. These all work. They're all kind of variations of decay in the soil food web. My only problem is that when you use KNF and things like this, you're producing word went right out of my head. I want to say methane. And on a large scale, I don't think that's good, obviously. On a smaller scale, I think it's okay. The stuff works. These tend to be anaerobic formulations that you're dealing with. There are anaerobic pockets in the soil. There is decay occurring in anaerobic situations. The byproduct tends to be methane. And that's my really only concern. But yeah, they work. They work, definitely.
And Pat notes, some have seen some harm from the GMO crops miles away, transmitted through soil to organic crops. What would you do? And I think probably the answer is more microbes, right?
Yeah, definitely. Yeah, GMO, the whole situation is, got to really look at the science here. Yeah.
Janet says, great talk. Thank you. She just ordered Microbes in Your Bacteria book.
Oh, super. Thank you. My publisher thanks you. Let me put it that way. As Lee will tell you, you don't make money being an author, but you do have fun.
Yep, you do have fun. All right. So with that, this has been a fabulous presentation. I'm just thrilled that you agreed to talk to us today, and so many great questions. And I just want to kind of highlight again the upcoming talks that we have later today. We will be talking with Tom Murray and Dr. Tom Dykstra in the next session. And then later on this evening, we will be talking with Juan Carlos and Robert from Sober Mesa on permaculture. And we were supposed to have Katie from the Savannah Institute on. Katie is ill, but hopefully we're going to have Eric from Savannah Institute kind of pop in for a few minutes just to kind of talk about what they've been doing in terms of polycultures. But all of these upcoming sessions kind of really tie back into what Jeff is talking about today. So this was a great setup for what we're going to see in practice. And again, like I said, Juan Carlos and Robert have taken what Jeff has written about and just kind of really put it to good use and good work. So you're going to get to see all of this in practice later tonight. So let's see if I should be telling you all something else. We will have this recorded session and all the other sessions up within a day or two of the presentation. So as members, you guys just log into the website with your email address and you'll be able to access that and any extra content that we have. I think Jeff is going to provide a couple of charts and things or pictures. I think he mentioned to Leslie. And other content related to the other sessions you'll be able to download. And then just on behalf of NAFEX, I want to thank Jeff for agreeing to do this. I know you're quite popular in terms of, probably being asked to speak a lot. So we appreciate you taking the time to do that. And if anyone has any, further questions, reach out to Jeff maybe through his website. It's jefflowenfels.com, right?
Jeff@gardiner.com.
Jeff@gardiner.com. Okay.
Yeah.
Yeah. So I think with that, we will end today. Leslie, have I missed anything?
Nope, you've covered it all. It was fantastic. Jeff, thank you so much for taking time to share your wisdom, because it certainly helps all of us as fruit growers.
Yes, and thank you all for attending today. And again, you'll be able to see this again. If there's anything you want to go back and re-listen to, by all means, log in a couple of days and find it and listen to Jeff again. So thank you again, Jeff, and thank you to all who came today. And thank you to Leslie for being my wing woman in the background. And we will see you guys at the next session, which is 5 o'clock this afternoon. All right.
Bye.
Bye.